Hello students, welcome to my channel. In this class we will see about the inverse PCR method. What is inverse PCR method and how it is used in the mutagenesis process. Let's see. Use only two primer. Here in inverse PCR there we use two primer, means two different primers. They are not similar in their sequence, and to create the desirable mutation. How we will see. Key feature is here, entire vector is amplified during the mutation. Entire vector will be amplified. Whole vector will be copy of their sequences, because in PCR whatever you provide, that will be amplified. So about the primer, we will see about the primer first. A pair of primer are used, means a pair means two in number primer are used, as we discussed already. The 5' termini hybridize end to end, sorry a minute. So 5' termini hybridize end to end on the opposite strand of the plasmid DNA. So this is the plasmid DNA, and here the 5' means this is 5' and this is 5' of the second strand, this is 3' and this is 3' of the another strand. Now it is saying this primer will be hybridized here at the 5' end. This is how of the plasmid. This is the plasmid and we making it in state form. So we can use in PCR. In PCR we cannot amplify the circular form of the DNA. That's why whatever type of DNA we have to make it in linear form. So here only one primer contain desirable mutation. What does it means? It means either this one or either this one. Only one is able to have it can't be able to, we make it to have single mutation primer. So we consider this one, we taking a mutate one. Okay. Primers are extend in opposite direction on the circular template DNA strand. That one was circular, so this is, now it will make or extend in their opposite direction, means opposite to each other. This will, the direction of this one is this and direction of this one is this. So they are extending in opposite directions. So now in detailed procedure we will understand the better what is happening here. In PCR amplification, entire double strand of the plasmid we use to amplify. Okay, then produce a linear double strand DNA, and that plasmid will be form a linear strand and contain mutated one end. So one end here is mutated and that will amplify more. Then at the end of the PCR, and if we are using 30 cycle of the PCR, then all maybe approximately most of the, no, half of the, because we use only single primer as a mutant. So half of the product is have and is mutated, because we use mutated at the 5' end as a primer is hybridized at the 5' end. That's why it's, we find the one end of the any DNA, half of our product is mutated. So the linear PCR is then circularized. Now after the PCR production is done, what we do circularization of that strands with the help of ligase. Ligase will attach any nearby present strand. So if this is linear, so we make it to circularize and then attach these ends with the help of ligase enzyme. Then circularized DNA is transformed into the E. coli. E. coli is bacteria where we transform our modified circularized DNA strand or plasmid. So what is the feature of this inverse PCR? Amplify the whole plasmid vector. Yes, and several variation because of many cycle of PCR we get so many strand which is mutated, which is not wild now. So this method is improve the efficiency of the mutagenesis. This is how, by the amplification, by the copy of the number of mutation, we get so many mutation here. This is all about the inverse PCR. In next class, we will see random and extensive mutagenesis. How random and extensive. We'll see in next class. Okay, bye-bye. Take care.